Our next topic with molecular genetics related to prokaryotic processes is going to be protein translation. All right. Protein translation essentially involves taking that messenger RNA that was produced during the process of transcription, reading it, and making a protein. All right. Where protein translation is going to happen is going to be at the ribosome. All right. And that's what I've got drawn here. All right. This is the large subunit of the ribosome. This is the small subunit of the ribosome. All right. One of the things that I need you to understand is that there is a difference in prokaryotic ribosomes and eukaryotic ribosomes. All right. If you notice, I've got two numbers here. I've got 70S and I've got 80S. All right. The S there is known as a Svedberg unit. All right. And essentially what it is a unit of measurement. And what happens with Svedberg units, you're taking proteins and you're centrifuging them at a high rate, so you're spinning them at a high rate. Those proteins will settle at whatever their molecular size is. All right. And in terms of that, the prokaryotic ribosome is slightly smaller than the eukaryotic ribosome. All right. And that's really important because this is how we can take antibiotics that target basic processes like translation, and it inhibits translation in the bacteria, but it doesn't inhibit translation in your eukaryotic cells. That is the critical difference, because you don't want to inhibit your own protein synthesis, because if you do that, your body will shut down. What you want to do is just shut down the bacteria. The difference in the ribosome is what allows that to happen. All right. Overall, with protein translation, a ribosome is always composed of a large subunit and a small subunit. I'm not so worried that you know the sizes. You will notice in your textbook that the size of the large subunit and the size of the small subunit don't mathematically add up to 70. Don't let that throw you. That's not an error. It has to do with the Svedberg units, okay? If you're taking the mass of something as a whole, this is going to be smaller than the two parts because of the surface area associated with the two smaller parts, okay? So in terms of that, no, prokaryotic ribosome is 70S, eukaryotic ribosome is 80S. What I want you to understand, in that large subunit, you've got an A site and a P site, all right? That A site is the acceptor site. That's where our transfer RNA is going to come in. It's going to bring in our amino acid so that we can start making our protein. P site is going to be where that tRNA will move to. Once you move to the P site, that's going to start essentially elongating our protein chain. So what's going to happen here, our ribosome is going to detect our RNA molecule here. And we'll have this going 5' to 3'. With RNA, usually that first codon to tell that ribosome where to start making the protein is going to be an AUG. All right. So if we feed our RNA in here, we've got an AUG sitting in there. What's going to happen? We're going to have a transfer RNA, and this is going to be the structure that kind of looks like a hairpin. All right. This is our transfer RNA. The role of that transfer RNA is to bring in our amino acid. So attached to it, it's going to have a methionine, because methionine is what corresponds to that AUG. And what it's going to do is it's going to sit in that acceptor site And on the tRNA molecule, I'm going to draw another one here just so you can see it. All right? We're going to have a region that's called the anticodon. So essentially what it's going to do, it's going to be the complementary sequence to what's in our messenger RNA. So if we've got an A here, we'd have a U. If we have a U, we'd have an A. With a G, it would be a C. All right? So that's how we're going to match up the correct tRNA to bring in the correct amino acid. Okay? Because in terms of that, you'll notice in the PowerPoints, I've got what I call it a super secret spy code breaker when I teach 1410, but really it's that table that lists the amino acids and the codons that go along with each amino acid. All right? So in this case, our AUG, this is going to be our codon. Essentially what it is it's 3 base pairs that will specify what amino acid is going to be inserted. All right? I want you to understand how to use the table. I do not expect you to memorize that table. All right? If I want you to be able to translate a sequence, I will supply you with the table. I want you to just be able to read it. Okay? So in terms of that, the AUG would correspond to a methionine. That would start our process of translation. What's going to happen? That RNA molecule is going to move between our large and small subunit, and it's going to carry that tRNA into the P site. All right? And this is where we're now going to start building our protein chain. Because once we've moved into the P site, all right, we now can have another tRNA molecule that comes in that's going to bring in a different amino acid. All right? And what amino acid is brought in is going to depend on what the codon is. All right? So in terms of that, we would bring in our next amino acid. And what will happen? That amino acid now will be connected to our first amino acid here. That's going to be what's going to start growing our protein chain. All right? So in terms of that, as the RNA moves, then this tRNA will fall away. Some books have in there, they've got another site in here. They call it the T site or the termination site. It's just where the tRNA kind of falls off. All right? That next one will move in, and our protein chain will keep elongating. All right? And it will keep reading that protein until it encounters a stop codon. All right? And in that table, you've got 3 codons that represent stop codons. What happens? A tRNA sits in, but it doesn't have an amino acid attached to it. That signals to the ribosome that it needs to let go of that protein chain because the protein synthesis is complete. All right? So in terms of that, stop codons are going to be important for ending the process. The AUG is your start codon. That's going to tell you where to start the process. All right? And really one of the things that's unique about bacterial translation and transcription, because there's not a nucleus in the prokaryote, both RNA transcription and protein translation are occurring in the cytoplasm. As soon as that RNA molecule comes off of the DNA, ribosomes latch on and start making proteins. So what you'll see in one of the images I have in the PowerPoints is of essentially a long chain of proteins that are being synthesized off of a single RNA molecule. Because as soon as that opens up, the ribosome will sit on, it'll start chugging out proteins. All right? So in terms of that, prokaryotes are a little bit more streamlined in going from transcription to translation. Eukaryotic cells, transcription is going to happen in the nucleus. That RNA moves from the nucleus into the cytoplasm. Cytoplasm is where translation occurs in eukaryotes. Prokaryotes, both transcription and translation are occurring in the cytoplasm because prokaryotic cells don't have a nucleus. All right? Hopefully some of that makes sense. Again, I've got some videos that are better with the 3D animations than what I can put up on the board. So take a look at those. If you have questions, please let me know.